Hello everyone, my name is Kary Mullis and I'm truly honored to be invited to this science forum today. I'll be talking about PCR, what it is, how it works, and the profound impact it has had on both science and society. Polymerase chain reaction is a technique used to make numerous copies of a specific segment of DNA quickly and accurately. The polymerase chain reaction enables investigators to obtain large quantities of DNA that are required for various experiments and procedures in molecular biology, forensic analysis, evolutionary biology, and medical diagnostics. PCR was fully developed in 1983. I won the Nobel Prize for Chemistry in 1993 for this invention. Before the development of PCR, the methods used to amplify or generate copies of recombinant DNA fragments were time consuming and labor intensive. In contrast, a machine designed to carry out PCR reactions can complete many rounds of replication, producing billions of copies of a DNA fragment in only a few hours. The PCR technique is based on the natural processes a cell uses to replicate a new DNA strand. Only a few biological ingredients are needed for PCR. The integral component is the template DNA, that is, the DNA that contains the region to be copied, such as a gene. As little as one DNA molecule can serve as a template. The only information needed for this fragment to be replicated is the sequence of two short regions of nucleotides, which are the subunits of DNA at either end of the region of interest. These two short template sequences must be known so that two primers, which are short stretches of nucleotides that correspond to the template sequences, can be synthesized. The primers bind or anneal to the template at their complementary sites and serve as the starting point for copying. DNA synthesis at one primer is directed toward the other, resulting in a replication of the desired intervening sequence. Also needed are free nucleotides used to build the new DNA strands and a DNA polymerase, an enzyme that does the building by sequentially adding on free nucleotides according to the instructions of the template. PCR is a three-step process that is carried out in repeated cycles. The first process is denaturation at 95 degrees Celsius. The second stage is annealing at 55 degrees Celsius, and the third process is synthesis, also which is also known as extension, at 72 degrees Celsius. The initial step is the denaturation or separation of the two strands of the DNA molecule. This is accomplished by heating the starting material to temperatures of about 95 degrees Celsius, equivalent to 203 Fahrenheit. Each strand is a template on which a new strand is built. This causes the double-stranded DNA to separate into two single strands. In the second step, the temperature is reduced to about 55 degrees Celsius, 131 Fahrenheit, so that the primers can anneal to the template. The short synthetic primers attach two complementary sequences flanking the target DNA region. These primers are crucial for ensuring the specificity of the amplification process. In the third step, the temperature is raised to about 72 degrees Celsius and the DNA polymerase begins adding nucleotides onto the ends of the annealed primers. At 72 degrees Celsius, the Taq polymerase is developed. It is an enzyme derived from the thermophilic bacterium. It synthesizes new DNA strands extending from the primers to match the target DNA sequence. At the end of the cycle, which lasts about 5 minutes, the temperature is raised and the process begins again. The number of copies doubles after each cycle, usually 25 to 30 cycles produce a sufficient amount of DNA. This exponentially amplifies the DNA, creating millions of copies of the desired sequence in a relatively short time. Key innovations such as the use of the heat-resistant Taq polymerase eliminates the need for manual enzyme injection, and the use of primers ensures precise amplification. In the original PCR procedure, one problem was that the DNA polymerase had to be replenished after every cycle because it is not stable at high temperatures needed for denaturation. This problem was solved in 1987 with the discovery of a heat-stable DNA polymerase called TAC, an enzyme isolated from thermophilic bacterium. This inhabits hot springs. TAC polymerase also led to the invention of the PCR machine. Because DNA from a wide range of sources can be amplified, the technique has been applied to many fields. PCR is used to diagnose genetic disease and to detect low levels of viral infection. In forensic medicine, it is used to analyze minute traces of blood and other tissues in order to identify the donor by his genetic fingerprint. The technique has also been used to amplify DNA fragments found in preserved tissues, such as those of a 40,000-year-old frozen woolly mammoth or of a 7,500-year-old human, which is found in a peat bog. When I first started working on PCR, funding was a significant hurdle. The potential of the technology was clear, but convincing others to invest in it was a challenge. Initially, my work was funded by the CETUS Corporation, but they had to choose whether to support PCR over other technologies. It was a crucial decision that eventually paved the way for PCR's development. Additionally, in the early days, PCR was far from perfect. The amplification was slow and the accuracy was questionable. The real breakthrough came with the discovery of Taq polymerase, a heat-stable enzyme that made PCR more efficient and reliable. Without Taq polymerase, we wouldn't have been able to overcome these technical barriers. Thank you all for your time and attention. I now welcome any questions from the audience and look forward to the discussion.
Thank you, Dr. Mullis, for that insightful presentation. Your research has clearly made a significant impact, and I'm sure the audience has learned a lot from your findings. Now, I have a few questions to further explore the implications of your work. First, how does your discovery compare to modern technologies? Second, were there any ethical concerns raised about your work? And if so, how did you address them? Third, how has your work influenced current biotech research? And finally, what advice would you give to young scientists today? We would love to hear your thoughts on these.
PCR remains a foundational technology in molecular biology, but advancements such as digital PCR and real time PCR have improved accuracy, sensitivity, and quantification. Digital PCR allows absolute quantification of DNA without needing reference standards, while qPCR enables real time monitoring and DNA amplification. Additionally, next generation sequencing has expanded on PCR by allowing massive parallel sequencing of entire genomes. One ethical concern with PCR is its role in genetic privacy and forensic science. The ability to amplify DNA from tiny samples raised concerns about unauthorized DNA profiling and privacy breaches. Additionally, PCR's use in genetic testing brings up ethical dilemmas regarding consent, genetic discrimination, and the psychological impact of knowing one's genetic risks. Addressing these concerns requires strict ethical guidelines, informed consent, and legislation like the Genetic Information Non-Discrimination Act to prevent misuse. PCR has had a profound impact on biotechnology by enabling rapid DNA amplification, which is crucial for medical diagnostics, forensic investigations, genetic research, and infectious disease detection. It has played a key role in detecting viruses like COVID 19, assisting in cancer research, and supporting innovations in synthetic biology and gene editing. Without PCR, many modern biotechnological advancements, including CRISPR gene editing, would not be as efficient or accessible. I would encourage young scientists to remain curious and open to unconventional ideas. Scientific breakthroughs often come from thinking outside the box, as PCR did when I envisioned DNA amplification working like natural replication. Persistence is key, as many discoveries face skepticism before gaining recognition. Additionally, embrace collaboration across disciplines, as biotechnology increasingly integrates fields like AI and nanotechnology to drive innovation.
Today we had an insightful discussion with Dr. Kary Mullis about the revolutionary polymerase chain reaction technique. We explored how PCR has transformed biotechnology by enabling rapid DNA amplification, its impact on modern scientific advancements, and the ethical considerations surrounding genetic research. We also discussed how PCR has paved the way for innovations like digital PCR, real time PCR, and next generation sequencing, which continue to shape the future of biotech.
PCR was a game changer when I first developed it, and seeing how far it has come is truly remarkable. From medical diagnostics to forensic science and evolutionary biology, PCR has become an indispensable tool in countless fields. Looking ahead, I envision biotechnology continuing to break barriers, perhaps integrating AI to enhance genetic analysis, improving precision medicine, and making gene therapies more accessible. The future is bright for young scientists who are willing to push boundaries and think creatively.
Dr. Mullis, thank you for sharing your knowledge and vision with us. Your work has left an incredible legacy in science and medicine. We also want to thank our audience for their thought provoking questions and engagement. With that, we conclude today's forum. Stay curious, stay innovative, and keep exploring the wonders of biotechnology.